Hello, my name is Travis Monk. This is one part of a series of videos introducing biology, the study of life. This video will provide an overview of experimental design, or how to properly approach setting up an experiment using examples wherever possible. The picture above outlines the scientific method, a means for studying the natural world. Experimental design is part of the scientific method. Its purpose is to help formulate a hypothesis and properly design a controlled experiment. Two important steps found in the middle of the chart. Without utilizing experimental design properly, it would be impossible to collect legitimate data or to draw accurate conclusions that can be used to gain knowledge about the world. There are 7 important components to setting up a scientific experiment that will be described on the next few slides. This slide covers the first 2 components, the independent and dependent variables. In a given experiment, there should be one single thing that is modified by the scientist, and one particular thing that's being measured. The intentionally changed factor is called the independent variable, or IV. By changing just one factor in an experiment, the experimenter can get an idea of exactly what happens in response to that change. If more than one thing is modified at a time, there is no guaranteeing what is responsible for a particular change. The dependent variable is sometimes referred to as the measured variable. The dependent variable, or DV, is what changes in response to the IV, or independent variable, being changed. The graphic to the right provides an example of an independent and dependent variable. A scientist might want to change the liquid food that a plant gets and measure how that affects the height of a plant. The independent variable, that which is intentionally changed, is the type of liquid used. The dependent variable, what is being measured, is the height of the plant after a set amount of time. If you were to conduct an experiment to test the effectiveness of a painkiller, you would want to compare it to something. You may be testing the effectiveness of painkillers against one another, Advil versus Tylenol, or compared to no painkiller at all. With pharmaceutical trials, a placebo, or a substance such as sugar that has no therapeutic effect, might be used for a comparison. All of these specific circumstances involve one independent variable. The specific types or values of the independent variable are the control and experimental groups. The control group is the point of comparison for the other groups. No painkiller or a placebo would make most sense in this experiment. The experimental groups would be Tylenol and Advil, those different substances that are being tested against the control group. The graphic to the right shows the effect of different drugs, either calcium pills or a placebo, a sugar pill, on the percent of patients suffering a myocardial infarction or a heart attack over a period of months. In this experiment, the placebo would act as a control group. The title for a scientific experiment should include both the independent and dependent variables. Someone looking at an experiment or graph's title should have a really good idea of what was being tested without reading any further. In the graphic to the right, a title such as acids, bases, bacteria, oh my, might sound really witty or cute, but it doesn't really clearly describe what's being tested in the experiment. The effect of pH on the activity of the enzyme being produced by indicates the independent variable, the dependent variable, and leaves very little to question. A scientific hypothesis should not be, as you've probably defined it throughout your science education, as merely an educated guess. When universities conduct research costing tens of thousands of dollars that require federal grants for funding, they are expected to have a really good idea of what should happen in a given circumstance and why. One of the requirements is that a hypothesis must be falsifiable, or it must be able to be shown incorrect. While hypotheses can be rejected, they are never proven. They're just supported by data. Hypotheses are often written in a format that is if-then. So if you study more than an hour for a test, then your scores on that test will increase, would be an example of a hypothesis. The picture to the right shows the steps involved in the scientific method. As the image illustrates, forming, testing, and revising a hypothesis is a cornerstone for the scientific method. In a given experiment, there should be one independent variable, as we mentioned earlier. As many other variables as possible should remain constant, or the same. If more than one factor is varied in an experiment, whether intentionally or unintentionally, the cause of some change might be left to question. In the graphic to the right, an experimenter is manipulating 2 different variables, the pH, or the acidity or basicity of a substance, and the temperature of a solution, and looking at how fast an enzyme works. As the graphs illustrate, both of those variables play a large role in how quickly the enzyme works. If both of those two variables were modified at the same time, the results may not accurately depict the relationship between the independent and dependent variables. So maintaining lots of constants is important. That is the end of this video summarizing experimental design and some of its most important vocabulary. If you're interested in learning about any other specific biology concepts, please subscribe to my channel. Thank you.
In this video, we will be looking at experimental designs. The types of experimental designs include the completely randomized design, the randomized block design, and the matched pairs design. In a completely randomized design, each experimental unit is randomly assigned to a random group to receive a different treatment. Each unit in the same group will receive the same treatment, and at the end of the experiment, you will compare the results from each treatment. Show how a completely randomized design would be used in the following example. A researcher wants to conduct an experiment to determine which environment is best suited for studying: a library, in one's own room, or outside. A total of 30 university students volunteer to participate in the experiment. So we have 3 treatments: the library, in one's own room, and outside. Since we have 3 treatments, we will have 3 groups receiving the treatments. Since there are 30 university students, these 30 subjects will be randomly assigned into 3 groups to receive the treatments. Since there are 30 people, each group will have 10 people, and at the end of the experiment, we would compare the results from each treatment. Let's say that the researcher believes that gender has an effect on the results. In a case like this, we would use a randomized block design. A randomized block design doesn't immediately randomly assign experimental units to receive treatments. We first assign people into a block based on a characteristic that is expected to influence the response to the treatments. In this example, gender would be the blocking variable. So first, we separate our experimental units based on gender. One block will be for females and one block will be for males. There are 18 females and 12 males. Then, the experimental units in each block will be randomly assigned to 3 different groups to receive a treatment. So let's make a diagram out of this. We are looking at a randomized block design, and gender was the blocking variable. We started off with 30 university students, and we separated these students into 2 blocks. One was for males and one was for females. We determined that there were 18 females and 12 males. We will have the females be randomly assigned to 3 different groups, and each group will have 6 females, and each group will receive different treatments. For the males, we are doing the same thing, except we have 12 males, so we will randomly assign 4 males into each group. And at the end of the experiment, we will compare the results within each block. The last type of experiment we'll talk about is the matched pairs design. In a matched pairs design, we are only comparing 2 treatments by using the same or similar experimental units. If we are using the same experimental units, we could have an example like this. What type of gasoline is more efficient? Type A gasoline or Type B gasoline? 3 cars will be used in this experiment. Describe how a matched pairs design would be used. If we are using the same experimental units, then each experimental unit will receive both treatments. So we have 3 cars, and each car will receive the 2 treatments, one with Type A gasoline and one with Type B gasoline. We would use the same car for each treatment, and which treatment goes first will be random. So for example, we could drive with Type B gasoline first, then once all the gasoline is finished, we would fill up the car with Type A gasoline using the same quantity, and at the end of the experiment, we would compare the efficiency of the gasoline. There are cases when we can't use the same experimental units for a matched pairs design, and we would have to use similar experimental units. For example, a researcher is trying to determine whether or not sleep deprivation has an effect on test scores. Describe how a matched pairs design would be used. Well, we could try giving both treatments to the same experimental units like we did in the other example. In other words, we could have the same person write the test on 2 different days, one being sleep deprived and the other with sufficient sleep. The problem with this is that we can't have the same subject write the same test because they would know what's on the exam when they write it the second time, so that would be kind of unfair. Instead, we could start off by pairing up students with similar GPAs. So for example, the two students with the highest GPAs would be matched up together, and the two students with the lowest GPAs would be matched up together. Then, each pair will be split up and randomly assigned to one of the two treatments. So for a pair, one person will get the sleep-deprived treatment, and the other person will get a normal amount of sleep. Then, we would compare the results for each pair, and we will also compare all of the results. So for example, if we saw that the sleep-deprived people were consistently getting lower test scores than the people who got a regular amount of sleep, then we can say that sleep deprivation was actually the cause for a bad test score. So let's quickly recap. These are the experimental designs we talked about. In a completely randomized design, each experimental unit is randomly assigned to a random group to receive a different treatment. This is what the diagram would look like. Group N, or treatment N, means you can have any number of groups or treatments. A randomized block design first assigns people into a block based on a characteristic that is expected to influence the response of the experimental units to the treatments. Then, a completely randomized design is performed within each block. This is what the diagram would look like. Block N means that you can have any number of blocks that you want. For a matched pairs design, we saw two scenarios, one that uses the same experimental units and one that uses similar experimental units. Both of these involve the comparison of only two treatments. If you are using the same experimental units, each experimental unit gets both treatments. This is what the simplified diagram would look like. If you are using similar experimental units, we first pair up similar experimental units, then each pair is split up and randomly assigned to one of two treatments. This is what the simplified diagram would look like.